Hi, I'm Joseph Cox, and this is my digital story of using micro FTIR for the analysis of paint. For this analysis, the instrumentation consists of a microscope coupled with a FTIR spectrometer. This allows for each paint layer to be analyzed using FTIR. The FTIR spectrometer is composed of an interferometer. The interferometer divides a beam of light into two beams and then recombines the light after introducing a phase difference in one of the beams. The split beam is reflected off of two mirrors, one fixed and the other movable. The movable mirror creates the phase difference. When the beams of light are in phase, the result is constructive interference that increases intensity. Out-of-phase beams result in a destructive interference that decreases intensity. The resulting waveform is called an interferogram. This can be solved with the Fourier transform, the math of this equation. This equation is solved using a computer. FTIR microscopy provides information about the chemical structure of the evidence being examined. Fibers in paint rotate, bend, and vibrate in different modes when placed in infrared light. Here are some bond types of the functional groups shown here. Paint samples can be examined to identify the paint and compare a possible paint source. Using FTIR microscopy, the layers of the paint can be identified and compared. Using functional groups, binders, resins, pigments, and extenders can be identified. Here are some common absorption bands for paint binders and resins. Common absorption bands for paint pigments and extenders are shown here. If these bands are found in paint samples and the potential source, it helps show that the paint sample may have came from the source or a similar source. This could help corroborate a physical match of the paints. Each layer of the paint sample can be absorbed differently. Binders and resins will have different absorption bands than pigments and extenders. This also helps in identification of paint layers. Each layer of paint, primer, or binder will absorb differently and can be identified. Using this information when compared to a possible source of the sample, a comparison can easily be made and a linkage can be formed. The shape and thickness of samples are important when using the FTIR microspectrometer. The ideal sample is approximately 5 to 15 micrometers thick. Thick samples can be examined by cutting and flattening them prior to observation with the micro FTIR. The sample is then viewed under the FTIR microspectrometer. Absorption bands are noted in each layer, and layer colors are recorded. All this information can be used to compare a potential paint source. The paint source would then be observed and compared to the paint sample. FTIR microspectrometry can be very helpful in supporting mechanical fit matches of paint chips. A physical match can be enhanced by comparing the chemical components of the layers in matching paint chips like the one shown here. FTIR showing similar absorption bands in each layer would increase the likelihood that these paint chips are from the same source. Micro FTIR can be very powerful in paint analysis. It establishes a reproducible comparison of paint samples. It also allows for individual layers to be chemically identified. This technique takes paint analysis to the next level.